Hi, I'm George Hammond, conference producer at Technology Networks, and I'm here to introduce our final talk of the day, allosteric small molecule activation of PI3 kinase for cardioprotection and neuroregeneration. I'm incredibly pleased to welcome Dr. Bart Vanhaesebroek and Dr. Gong. Dr. Vanhaesebroek is a professor at the Cancer Institute, UCL, and is an elected member of EMBO and of the UK Academy of Medical Sciences. His laboratory has undertaken studies on the PI3 kinase signaling system that have defined mechanisms and physiological processes relevant to cancer, immunology disorders, and overgrowth syndromes that these enzymes control. His research has widely contributed to the development of multiple approved medicines. Whilst Dr. Gong completed her PhD at the University of Auckland, New Zealand, focusing on designing and characterizing small molecule inhibitors that target the PI3 kinase signaler. She then joined Professor Bart Vanhaesebroek's laboratory to work on small molecule modulators, PI3 kinase, and received a Mary Skłodowska-Curie Personal Fellowship to support this work. A very warm welcome to you both. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk. So feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during this presentation. And you can also benefit from some fantastic supporting literature, which you can download for free below the video player. And now, without further ado, I will hand over to Dr. Vanhaesebroek and Dr. Gong to begin their presentation.
Hi, welcome everyone. It's my pleasure to present this talk together with Dr. Gong. As you can see, we live and work in London, and we will talk about small molecule activation of PI3 kinase. So just add to what George said, here's a picture of Grace. Grace is currently seconded to Roger Williams' laboratory and the MRC Laboratory of Molecular Biology in Cambridge, where she is performing further structural studies. I am a basic scientist, and one thing we did that is probably memorable is discovery of PI3 kinase delta, which we took from gene cloning over preclinical modeling and inhibitor development to clinical approval of PI3 kinase delta inhibitors. So we are actually basic scientists, not full-blown drug development experts. We work in academia, but we hope that the audience will appreciate the work we have been doing. So just a bit of general background. Medicines are known to work by modulating activities in cells, and most drugs or medicines are blockers, inhibitors, although some activator drugs are very well known, and they are agonists and often artificial ligands of G-protein coupled receptors, GPCRs. Most of you know activators of beta-adrenergic receptors that are used to treat breathing conditions like COPD and asthma, and a lot in the news recently, GLP-1 receptor agonists, which are anti-diabetics and also give weight loss induction. Nuclear receptors are also being targeted by activators. These are ligand-activated transcription factors, and PPAR agonists are being used for metabolic diseases. So today's talk focuses on kinases. And drug development in the kinase area has focused almost exclusively on inhibitors to date. And but kinases, and so basically to block the kinases, but kinases have also a positive impact, so you may want to stimulate them. They can be involved in tissue repair and wound healing. You may want to stimulate kinases to give neuroprotection or neuroregeneration, protection from ischemia reperfusion, and so on. Basically to stimulate the metabolism and the immune system and so on. A key question really is it possible to generate small molecule kinase activators? And in fact, there are quite a number of proof of concept studies for multiple kinases like AMPK, which has been targeted quite extensively for metabolic sensitization, and a range of other kinases, but no approved kinase inhibitor drugs are currently available. Some general principles of kinase activation. Activators and inhibitors, we believe, are governed by distinct principles in terms of level of duration and target modulation. What does that mean? Inhibitors often you require sustained and full inhibition to be therapeutically effective, so as in cancer. A kinase is active in cancer, it drives the cancer, you really want to block it completely to get the cancer out of the way. However, with activators, even a transient or a partial kinase activation could be expected to be clinically meaningful. For example, you're going to have an insult, you give a kinase activator, you protect to some extent to this insult. If you want to stimulate wound healing, anything you get 10% activation, 30% activation, 100%, 200% activation will actually stimulate the regeneration better. So you tend to have a clinical impact, even not upon full activation and continuous activation. However, there are challenges to create kinase activators. Small molecule kinase activators target the ATP binding site. When it comes to activators who are targeting allosteric sites, the binding sites are mostly unknown. Also, when you activate kinases, this actually often releases autoinhibitory interactions, which makes the molecules a little bit more flexible, and that can create issues with structural studies, and you get insufficient structural resolution. High resolution is needed for subsequent medicinal chemistry approaches. However, there are new developments that can facilitate this unbiased screening for activator molecules. The libraries have really extended, including DNA-encoded libraries. There are assays that you can use to follow specific kinase conformations, and developments in structural biology like hydrogen deuterium mass spectrometry, which allow to look at interaction of small molecules, ligands with proteins in their intact state. And our talk will provide an illustration of some of these approaches. So let's now talk about PI3 kinase. Sorry, this. Okay, and just a little bit of background here for people who want to read more. In fact, there are very few reviews on kinase activators. As you can see, these are reviews more than 10 years old, and there's a recent review shown here. So what about PI3 kinase? We focus on a subset of PI3 kinases that are stimulated by growth factors, shown here, G protein coupled receptors. And what they do, they phosphorylate lipids in the plasma membrane. These lipids are getting a phosphate and then interact with target proteins, some of which are well known, like AKT, BTK protein kinases, adapter proteins, and so on. And these kinases have many substrates, and then they regulate a whole range of biology. So let's now focus on the PI3 kinases themselves. They consist of a P110 catalytic subunit, alpha, beta, gamma, or delta, and they are in complex with a regulatory subunit that allows them to engage with this receptor complexes. So the receptor, okay, let's go further down. What these kinases are doing, given that they are involved in so many processes, they are implicated in many common disease states like cancer, immune dysregulation, dysregulation of the metabolism, aging, neurobiology, cardiovascular. And some people are also born with germline mutation or develop these mutations early in life. And these are in rare diseases where you have overgrowth syndromes, PI3 kinase driven overgrowth syndrome, PI3 kinase delta, immune syndrome, and people are born with a gene that downregulates PI3 kinase, and they are predisposed to cancer. So given this multiple involvement in diseases, it's not surprising that pharma has developed inhibitors to PI3 kinase. And, this has actually been a massive effort. But because PI3 kinase have so many biological activities, very few drugs have been approved. But before I show these drugs that have been approved, just to recap the isoforms. PI3 kinase alpha, beta are ubiquitously expressed. They're often referred to by their gene name. Gamma and delta are expressed in leukocytes and immune cells and regulate the immune system. activating mutations in cancer are really only found to a high extent in the PI3 kinase alpha isoform. So here are the drugs that are approved. Alpha inhibitor from Novartis that is now also approved for this rare disease overgrowth. And then a range of drugs that are mainly targeting PI3 kinase delta on its own or together with other isoforms. And they are involved in approved for B-cell malignancies and this immune syndrome. So thinking about PI3 kinase activation, PI3 kinase activation could also do a lot of good things, and people have used genetic approaches and pharmacological approaches, often inhibitors, to show that PI3 kinase and PI3 kinase family members could be involved in all these positive effects as I show here. So the question really is, if PI3 kinase or PI3 kinase family members can be genetically activated, we ask the question, can this also be achieved pharmacologically? And so we went on to look for an alpha selective activator. We published this paper recently in Nature with Grace as the first author. This tool compound is available from Cancer Tools here. And so I will now hand over to Grace, who will take you to the high throughput screening, the hit confirmation, some medicinal chemistry, biochemistry, structure, activities in cells, and I will come back to talk to you about potential in vivo applications. Over to Grace now.
Okay. All right. So thank you, Bart, for the introduction and the background. So like Bart mentioned, we work on a protein called PI3 kinase alpha, and that consists of a P110 alpha catalytic subunit and a P85 regulatory subunit. So in the unstimulated state, P85 binds to and holds P110 alpha in an inactive conformation. So in order for the protein to be activated, there are 4 distinct events that needs to happen in the PI3 kinase alpha catalytic cycle. These can be briefly summarized into 2 points. Firstly, there has to be weakening of the inhibition between P85 alpha regulatory subunit and the P110 alpha catalytic subunit. Secondly, there has to be increased interaction of the enzyme with the lipid membrane where its substrate is located. So PIK3CA, the gene encoding the P110 alpha catalytic subunit, is actually one of the most frequently mutated genes in cancer. And these oncogenic mutants either mimic or enhance one or multiple steps of the events in this catalytic cycle. So this then led us to think if, like Bart mentioned, if PI3 kinase alpha can be activated oncogenically, can we also do it with small molecule activators? So to answer this question, Bart had set up a research consortium consisting of his own lab at UCL Cancer Institute, Richard Angel's group at the UCL Translational Research Office, Roger Williams at the MRC Laboratory of Molecular Biology, and Dave Smith and his Open Innovation team at AstraZeneca. So through combined efforts, we performed a high throughput screen where we screened approximately 500,000 compounds from the AZ library. Richard Angel and his group then performed some medicinal chemistry efforts to try and improve our hit compounds, and this yielded us our hit candidate UCL TRO 1938, which I will then refer to as 1938. So to look at the effect of 1938 on PI3 kinase activity, we performed a ADP glow kinase assay using recombinant protein. So we can see there, following stimulation by 1938, we saw an increase in the activity of PI3 kinase alpha. However, 1938 did not affect the closely related PI3 kinase beta and delta isoforms. Prior to the discovery of 1938, we often used a phosphopeptide derived from PDGF receptor as a positive control to stimulate PI3 kinase activity in vitro. So I've called that PY here. And that led us to think, what happens if we now try and co-stimulate PI3 kinase alpha with the phosphopeptide and 1938? So surprisingly, we actually saw this huge synergistic activation of the enzyme upon co-stimulation. Now, this is really interesting because at 1 micromolar, we are already using the phosphopeptide at saturating concentrations. So if we increase that from 1 to 10 micromolar, we don't actually get further stimulation of enzyme activity. So the fact that we have this huge synergistic activation tells us that although 1938 and the phosphopeptide may be sharing mechanisms of how they're activating the enzyme, 1938 is definitely doing something unique that this phosphopeptide is not. So then we also tested the ability of 1938 to activate oncogenic mutants of PIK3CA. The summary, just to summarize the results, 1938 was able to increase the activity of all of our mutants that we've tested. So looking at the crystal structure of P110-alpha co-crystallized with 1938, it gave us good insight into potentially why is 1938 so selective for the alpha isoform over the closely related beta and delta isoforms. So we can see that for the binding site in P110-alpha, there is a nice cavity here to accommodate the ligand binding. However, that same region in beta and delta, this pocket is actually blocked by amino acids. In addition, we also performed a kinome screen and screened 133 kinases. We did not see any significant protein kinase inhibition by 1938. We also tested the effect of 1938 on other PI3 kinase isoforms and other PIKKs, including mTOR and ATM, and also we did not see any effect on these kinases 1938. So here is a summary slide of our findings from some of our key structural work. We know that 1938 allosterically activates PI3 kinase alpha, like what we would expect. It binds outside of the ATP binding site, and the binding site is at the interface between multiple domains in P110-alpha. Interestingly, the binding pocket of 1938 is actually not present in the APOL protein. So the pocket is induced by 1938 itself. When 1938 binds, we observed multiple structural changes, including disruption of the inhibitory context between P85-alpha and P110-alpha. So coming back to what Bart and I mentioned earlier, P85-alpha regulatory subunit inhibits P110-alpha catalytic subunit and holds it in an inactive conformation. So in order for the protein to be activated, we have to disrupt these inhibitory contexts. We also saw changes in the active site, including repositioning of the kinase activation loop and also movement of the ATP loop towards the ATP binding site. So basically, 1938 activates PI3 kinase alpha through a distinct mechanism. It enhances multiple steps of the PI3 kinase alpha catalytic cycle, and it causes both local and global conformational changes in the structure of the protein. So now I would like to go into a bit more details of the specific structural studies that we performed. So firstly, we used hydrogen deuterium exchange to study the effect of 1938 on PI3 kinase alpha. This technique measures the exchange rate between hydrogens on the amide backbone of a protein with deuterium in solution. So it gives us an idea of the dynamics and movement of the protein's secondary structure. I won't go into this in a lot of detail, but just to say we identified a region in the C lobe of the kinase domain that was more protected or less flexible when the activator was binding, and this suggested to us to be the potential binding site of 1938. Now, if this was true, it would also mean that 1938 does not bind at the ATP site and it is allosteric. So I have a model of ATP there shown in yellow just to show you where the active site is. In addition, we also saw numerous regions that were less protected or with increased flexibility, and this suggested to us to be weakening of the inhibition that P85 has on P110-alpha. Again, this is critical for activation of the enzyme. We then turn to crystallography to look at the potentially important protein-ligand interactions and also exactly where the compound is binding. So on the left-hand side in orange, I have a model of an ATP competitive inhibitor to show you where the active site is. The partially disordered activation loop is shown in yellow. 1938 is shown as the magenta spheres, and in blue was the region that was identified by the HDX as being more protected upon activation. So we can see that the crystallography data and the HDX data is in good agreement with each other. The crystal structure also gave us good insight into some of the potentially important protein-ligand interactions. Of these, perhaps the most important are some hydrogen bonding interactions with an aspartic acid residue. We know this is important because if we modify the compound so that it can no longer make these hydrogen bonding interactions, the compound becomes inactive. In addition, there is another phenylalanine residue that potentially makes some face-to-edge interactions with the compound. And again, if we modify the compound so that it can no longer make these face-to-edge interactions, the compound becomes inactive. The crystal structure also gave us some good insight into the global conformational changes that occur upon binding of 1938. So again, 1938 is there shown in the magenta spheres. Close to the binding site of 1938 is a helix here, which I have colored in purple. We called this the PRD-like helix because this is analogous to the PIC regulatory domain that is found in PIKKs such as mTOR and ATM. And this is a regulatory site important for enzymatic activity. So we can see that when 1938 binds, this PRD-like helix moves towards the activation loop. Now, the activation loop is partially disordered in this structure, but in structures where it is complete, we know that the activation loop packs against this PRD-like helix. So it is possible upon binding of 1938, the PRD-like helix moves towards the activation loop. This then causes the activation loop to move closer to the ATP binding site, potentially adopting a conformation that is more suitable for phosphate transfer, hence increasing the enzymatic activity. Another change we observed was that when 1938 binds, the helical domain, which is here colored in green, pivots and moves the ATP loop closer to the ATP binding site. And again, this can facilitate better phosphate transfer. Thirdly, we saw movements across multiple domains, including the helical domain in green, the C2 domain in cyan, and these, together with the HDX data, would suggest to us that upon binding of 1938, there is a weakening of the inhibition that P85 has on P110 alpha, which I've mentioned before is critical for activation of the enzyme. Lastly, I would like to just point out that close to the binding site of 1938, we actually found a cluster of cancer-associated mutations here shown in yellow. Perhaps of the most important are two hotspot mutations here, E542 and E545K in the helical domain, because this region is one of two of the most frequently mutated regions in the protein. So the crystal structure allowed us to identify some potentially important protein-ligand interactions. So in order to figure out whether these amino acids are indeed for binding of 1938 and for the activity of 1938, we performed mutagenesis with a combination of these amino acid residues. So here is another in vitro kinase assay, and we can see that unlike the wild type, which responded nicely to stimulation by 1938, none of these other mutants did. However, we know that these mutants have retained their basal kinase activity, and they can also be further stimulated by the phosphopeptide control. So they're not kinase dead, they're just resistant to 1938. And this also tells us that the residues we have identified here are indeed very important for 1938 activity. The crystal structure also helped us to understand some of our earlier structure-activity relationship data when we tried to modify the compounds and figure out which part of the molecule contributed to activity. So we broke the compound into 3 regions. There is the blue region consisting of the acetylindolene, the orange region with the pyridine core and an amine, and the yellow region consisting of the piperazine. So our 2 analogues here of 1938, 1887 and 1899 had modifications in this yellow region where the piperazine was modified. Now, both of these compounds had activity that is comparable with 1938, with 1887 at 80% and 1889 at 102% relative to 1938. So this tells us that modifications in this yellow region are quite well tolerated by the enzyme. However, if we remove the piperazine completely, this compound is actually inactive. Now, this may partially be due to the fact that this compound had poor solubility. If we modify the blue region and change the indolene into a pyrimidine, the compound is actually inactive. And looking at the crystal structure, this may be because with this change, this compound no longer makes favorable face-to-edge interactions with this phenylalanine residue, and so the compound becomes inactive. If we modify the core and change the pyridine into a pyrimidine, the compound is again inactive. Now, this is quite interesting because we know at physiological pH, this nitrogen here is actually predominantly protonated. This means it can make some hydrogen bonding interactions with the aspartic acid residue. However, if we change the pyridine into the pyrimidine, as in this analog, this nitrogen here is no longer protonated at physiological pH, meaning that it can no longer make the same hydrogen bonding interactions with the aspartic residue that is required for activity. This also explains to us why 1938, given that it has a typical kinase scaffold with a pyridine and an amine group, why is it not a kinase inhibitor? Because if this nitrogen is predominantly protonated, that does not allow 1938 to make the typical donor-acceptor type interactions with the hinge region and kinases. Therefore, although 1938 looks like it could be a kinase inhibitor, it actually is not a kinase inhibitor. So moving on to the next section, I would like to talk a little bit about the effect of 1938 in cell signaling. So in order to look at the effect of 1938 on PIP3 production, which is the product of PI3 kinase alpha in cells, we use live imaging of the A549 cells expressing a fluorescent biosensor for PIP3. So we can see that upon stimulation of 1938, stimulation of the cells with 1938, there was a rapid and acute increase in the production of PIP3 at the lipid membrane in the wild-type cells. These are shown in the black traces. When we add in the BYL719 inhibitor, which is an inhibitor selective for PI3 kinase alpha, we can acutely neutralize the signal. However, in the knockout cells in red without the P110 alpha, we did not see any stimulation of PIP3 production by 1938. Now, this is demonstrated in a more visual way in this panel here below. We can see that in the wild-type cells, when we stimulate them with 1938, there is increased PIP3 production at the lipid membrane. And when we add in BYL719, the inhibitor, we can neutralize this effect. So AKT is a downstream effector of PIP3, and looking at the phosphorylation of AKT is a common readout for measuring PI3 kinase signaling. So here we did some Western blotting in mouse embryonic fibroblasts. Again, these cells are either wild type for P110 alpha or knockout for P110 alpha. In the wild type cells, we had a nice concentration dependent induction of phospho-AKT by 1938. However, in the knockout cells, when the target is not present, we did not get any phosphorylation or increase in phosphorylation of phospho-AKT. So this would tell us that the increase in phospho-AKT that we see here, induced by 1938, is due to specific activation of P110 alpha. Now, because measuring phospho-AKT is commonly used as a readout of PI3 kinase signaling, this is a type of biased analysis. We then turned to phosphoproteomics, which is an unbiased method of measuring cell signaling. We induced these MEFs with 1938 either wild type or knockout, and we can see that there was upregulation here shown in red and downregulation shown in blue of many phosphosites across many proteins. Now, most of these targets are known to be regulated either by the PI3 kinase signaling pathway or by the insulin signaling pathway. However, we did also identify some new targets which were previously unassociated with PI3 kinase signaling. Now, in the knockout cells without P110 alpha, we did not see either up or down regulation of these same targets. So I presented in a previous slide that we managed to create some 1938 resistant mutants using recombinant protein. And then we thought, could we repeat this in a cell model? So again, using MEFs that are knockout for PI3 kinase alpha, we induced these cells, we transfected these cells either with the wild type or with some of the mutants that we previously identified. So interestingly, if we restored the wild type back into these knockout cells and stimulate them with 1938, these cells, which now has P110 alpha, becomes responsive again to 1938. If we transfected the mutants which were resistant in the in vitro assays, we did not get any induction of phospho-AKT by 1938. So then we next moved on to look at the kinetics of 1938 induced signaling in the A549 cells, again using phospho-AKT as the readout. So we can see if we induce these cells with 1938, phospho-AKT signal is strong for about 6 hours, and then at 24 hours, there's actually dampening of the signal. We see the same kind of kinetics with insulin as well, and this is shown in a bit more detail on the graph on the right-hand side, when there is rapid induction of phospho-AKT following stimulation by either insulin or 1938, the signal is sustained for a couple of hours, and then at 24 and 48 hours, the signal decreases. Now, because insulin and 1938 follows such similar kinetics in the cells, this would suggest to us that the signaling induced by 1938 is also sensitive to the regulatory mechanisms within the cell. It tells us that the PI3 kinase signaling induced by 1938 is transient, and the signals induced by 1938 is not the same as those induced by the oncogenic PIK3CA mutants. So PI3 kinase alpha is well known for its ability or its role in cell proliferation and cell cycle progression. So then we tested the effect of 1938 on cell viability using a cell titer glow assay. Now, in the mass that are wild type for P110 alpha, there was a nice increase in cell viability following treatment with 1938. This was not seen in the knockout cells. At the higher concentrations, we saw a decrease in cell viability across both cell lines, and this suggested to us to be potentially specific toxicity of the compound. And then to investigate whether this increase in cell viability is actually due to an increase in active cell proliferation, we did some EDU assays and looked at the percent of cells in S phase. So again, in the wild type cells, when we stimulate them with 1938, there is an increase in the percentage of cells that are actively proliferating. This could be reversed by adding the BYR719 inhibitor. And in the knockout cells shown in gray, there was no increase in the cells in S phase. So now I will hand over back to Bart, who will talk about the therapeutic potential and applications of pharmacological PI3 kinase alpha activation.
Okay, thank you very much, Grace. That was very clear. So we next asked the question, what can we do with the pharmacological PI3 kinase activator in a therapeutic context? The first context we looked at is to protect the heart from acute insult when you reperfuse after an infarct. The other one is regeneration of nerves. And so we did mention that PI3 kinase activation can be implicated in cancer. So there is, the slight worry that chronic treatment with the PI3 kinase activator may lead to cancer, although we think because it signals very much like growth factors, this is not the case. But what we initially went for are conditions that allow us to stimulate short-term with the PI3 kinase activator, and we only give local administration of the activator and not systemic. So the first one is ischemia reperfusion injury. So let me give you a bit of background here. So we have a heart, the coronary arteries are blocked, and certainly there is ischemia as a consequence of oxygen starvation, and that gives damage. Remarkably, if you reperfuse with oxygen, this leads to what's called reperfusion injury, and you get actually more damage. Oxygen is a dangerous friend. It induces reactive oxygen species and so on. And so in order to have good recovery, it's important to limit infarct size. And so it's important to actually reduce the damage induced by this clinically relevant reperfusion, which is done with the stent. So the heart can protect itself. There are the so-called cardioprotective signaling pathways, which have been called reperfusion injury salvage kinase pathways, the risk pathway. It's a group of pro-survival kinases which confer cardioprotection when you activate at the time of reperfusion. And they can be stimulated by insulin, growth factors, and so on, as shown here. And they basically stimulate the MAP kinase pathway and the PI3 kinase pathway, giving rise to still not fully understood pathways of cell protection. One can also do this by a protocol called ischemic preconditioning. What you do is, for example, in the arm, using a tourniquet, you block flow to the heart and you induce several short cycles of non-injurious ischemia. So the heart senses that there is oxygen starvation, and basically you switch on these pathways using this ischemic preconditioning protocol, which we call IPC. And that protects you from a sustained ischemic insult. And so IPC switches on this pathway. So in previous work done by our collaborators Sean Davidson and Derek Yellen, they looked at IPC and what would happen if you added PI3 kinase inhibitors at the same time. I won't go into detail, these data are published. So here you look at the infarct size of the heart, you have a certain damage. When you do IPC, you actually have a reduced damage, and it's very clear when you together with this IPC gives a PI3 kinase inhibitor, either one from Genentech or Novartis, that this protective effect is no longer seen. So the question is, if a PI3 kinase inhibits this pathway, can we actually, using a PI3 kinase activator, induce this protection? And yes, we can. The activator does protect against this model of cardiac ischemia reperfusion injury. This is shown in explanted hearts here. So this is a heart cut in slices from the top to the bottom. This is stained with tetrazolium. The red is live tissue, the white is infarcted dead tissue. And it's very clear that, as is quantitated here, that a short-term infusion of 1938 for 15 minutes gives a very substantial activation, protection against insult, which correlates with phospho-AKT activation. And this is done in explanted hearts. We also have data in live animals. So the next one is regeneration of nerve injury. So why would the PI3 kinase activator stimulate nerve regeneration? Well, growth factors are known to trigger regenerative signaling. Nerves do regenerate, at least the peripheral nerves. And so this is happening through growth factors, nerve growth factor, and so on. They have multiple signaling pathways, but as you see, PI3 kinase is one of the common teams here. So because it's a common signaling pathway in regenerating neurons, we thought, why don't we, instead of using 1 or 2 growth factors, why don't we try to short circuit this and basically, pump up the signal and use a PI3 kinase activator. So just to recap, peripheral nerves do regenerate naturally. The problem is really that it is too slow. And the nerves do regenerate muscles. If the muscles are not innervated, they degenerate. And, this gives rise to problems with movement and so on. And there are currently no agents to stimulate nerve injury. So did we have some evidence for this? Yes, we first did an in vitro model for neuroregeneration. This is explanted rat dorsal root ganglion cultures, which is a mix of nerves and other cells, neurons and other cells. And basically you see that the PI3 kinase activator in a dose-dependent manner and a PI3 kinase alpha-dependent manner induces neurout outgrowth, which is a proxy for nerve regeneration. So these data stimulated us to do an in vivo model for neuroregeneration. And this is the sciatic nerve crush model in the rat. So here is a rat, you have the spinal cord, you have the sciatic nerve that innervates a muscle. And what you can do, you can actually give induce damage, for example, a crush, as you can see here. The crush site is shown here. Then you can inject a 1938, and also in initial experiments, we did these experiments with a pump to release 1938 over time. And then you can actually measure this is the nerve shown here. You can actually measure regeneration by histology. You can see how the nerve interacts with the muscle. You can do electrophysiology. Basically, there's a lot of assays you can do to monitor the impact on regeneration. I think I show here administration of 1938 with the pump. In fact, subsequent experiments show that we don't need to do that. Just a single injection does the trick for us. So analysis 3 weeks after injury, we have the muscles are better innervated. We have also electrophysiological evidence that things are working better in the nerve. And so, I will not, you can look at this in your own time at all these technical details that indicate that things are regenerating well and quicker. The mechanism, as far as we can tell, that all our observations are compatible with what the 1938 does. It simply accelerates the natural neuronal generation. We were initially a bit worried that this PI3 kinase activator would lead to sprouting of the nerves and so on. This is actually not the case. This looks like perfectly normal. It just happens quicker. And you can see that, speeding up nerve regeneration to contact the muscles quicker, keeping the muscle intact is obviously a key desire of the regeneration. And we are convinced that there is actually commercial opportunity in here, and we are currently seeking investment to take 1938 into the clinic in peripheral nerve injury. So time to summarize. 1938 is the first small molecule allosteric activator of PI3 kinase alpha. It's selective for PI3 kinase alpha over other family members of the family. It causes conformational changes in the PI3 kinase protein structure and has a unique mechanism of activation. It induces PI3 kinase alpha-dependent signaling and biology in cells. It has potential in tissue protection and regeneration. And for basic scientists who want to explore PI3 kinase isoform signaling after inhibitors and genetic knockouts, we now also have a tool to activate PI3 kinase alpha specifically. And overall, our work illustrates that it is possible to develop high-quality small molecule kinase activators. A visual summary of our work is shown here. We published a review paper in 2009, where we looked at how PI3 kinase was regulated. And so we got the cover of this TIPS review. And this gentleman here is advocating to stop PI3 kinase. I think times have moved on. What we suggest we should do on top of stopping PI3 kinase is also activate PI3 kinase. So here are the acknowledgements. A lot of people have been involved. We will leave this slide up for you to look at. I would like to particularly thank the EU Marie Curie EU funding to support GRACE, funding from the MRC, BBSRC research councils. We also received substantial funding from the NIHR, which is the NHS research funding from the UK, a fantastic collaboration with AstraZeneca. My lab is also supported by Cancer Research UK. Grace and myself are available online to answer any questions you may have. Thank you very much for your attention.
Thank you very much, Bart and Grace, for that fantastic talk. We are now moving to the Q&A section of these sessions, and I am aware of the time, so let's dive straight in with our first question directed for you both. Why did you start this work, and what was the rationale behind it, given that the field has traditionally been focused on kinase inhibition?
Do you hear me?
Yes.
Do you hear me now?
You, Bart?
Oh, sorry about that. I have too many. The rationale, well, to be absolutely honest, we try to express PI3 kinase mutated PI3 kinase in cancer cells, and this does not work. They don't tolerate it. So, as a cancer researcher, I thought instead of making PI3 kinase inhibitors, maybe I should try to make PI3 kinase activators. And in unpublished work, we have actually shown that we can kill cancer cells with an activator. But that is work for the future. There is more interest now in exploiting signaling overactivation as a therapy in cancer. So our initial ideas came from cancer, overactivation and signaling in cancer. I think with regards to the activator we have now, I think this is way too early. The compound is not fully optimized for this type of purposes. So we are focusing on short-term activation and local administration of this compound.
Thank you both. And now let's move on to your second question. Grace, you mentioned that 1938 is selective for PI3 kinase alpha over other isoforms and kinases. Could you discuss the implication of this selectivity in potential therapeutic applications?
So this can, with 1938 being so selective for alpha over the other isoforms, it could help us to regulate alpha-specific signaling and avoid adverse effects of, for example, overactivating all the isoforms at once. It could also help us to use 1938 as a tool to try and understand alpha signaling in biology, for example, in cells. So yeah, I think the biggest effect is it would help us avoid adverse effects of activating all the isoforms.
Thank you. Just to add in on this question from Nur Eliza, if you have already identified mutations, how do you then justify continuing using 1938 clinically?
I'm assuming this is referring to the mutants that's resistant to activation by 1938. So we have tested two different lots of mutants. So one of them are these mutants that were resistant to 1938. Now these were deliberately made in the hope that we could abolish activity of 1938. This is really to show that 1938 is binding, where the crystal structure is showing it's binding, and these interactions that we're picking up from the crystal structure are indeed important. So with these mutants, we wanted to make them resistant. In terms of naturally occurring mutations at these particular spots, I believe on the cosmic database, there are very few frequencies of these mutations, less than 10, I believe. So I don't think that would be a problem of using 1938, moving it further into the clinic. With the oncogenic mutations that occur naturally in 1938, this was presented in one of the earlier slides. So I tested 4 oncogenic mutants, including 2 hotspots. And just to put that into perspective, compared to, say, less than 10 on cosmic for these resistant mutations, the hotspots, I think we are over 6,000. And for both of these hotspot mutations and 2 other oncogenic mutants I tested, 1938 was able to stimulate them still.
Thank you. And as we are now drawing to a close for this session, I'd like to wrap up with one final question. Does 1938 have sufficient resistance to enzymatic metabolic clearance in order to have a pharmacological effect in vivo?
Okay, that's an interesting question. Look, this is an academic drug development program, so we have not really optimized this compound. After the paper, we got some further investment, something we are doing. I think this compound is not very stable in circulation. And in fact, that's what we at this point want, because if we go for short-term treatment, should there be some leakiness in the, let's say, or exposure in the blood, we'd rather have the tissue, the effect staying local and not going systemic. I can tell you that we have dosed this mice for 5 weeks with this activator. We do have exposure in vivo. We do see phospho-AKT, so signaling pathway activation. So there is some, let's say, biological activity seen, and this is given by gavage or by incorporation in the food. But we really feel that this is an early stage compound, and we want to, make this compound more potent. Now that's a micromolar potency compound. We really would like to go to a nanomolar compound. But your question is well taken, and I think the fact that it is maybe not staying long in the circulation is actually, for the purposes we have in mind, a good thing.
Thank you both so much. Unfortunately, that's all the time that we have for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thanks again, Bart and Grace, and we'll be back tomorrow at 8 a.m. Pacific time for another fantastic lineup of talks. Goodbye.
Thank you.
Thank you.
Bye.